Okay. So welcome to our 25th lecture. So today we're going to wrap up the module on machine learning. So today we're going to talk about foundational and other kind of frontier techniques in AI models, such as generative approach and their application in biology. So today's agenda, we're going to talk about how we change from the usual supervised and unsupervised learning based approach to a generative approach, and how are the generative model designs, what kind of assumptions they have, and how do we apply them in biological problems. And then we're going to talk a little bit about the concept of foundational model, which is a type of model training that you do so that you can use the model on different data sets and be utilized by other laboratories. Okay, so we switched to a generative approach because if you compare the usual type of classification, the regression model that we usually see, in order to classify sample, the model only need to learn this dash line. It doesn't need to know anything else about the data. It just needs to know that this dash line can perfectly separate data into groups. So it's trying to learn a very simple solution. But if we want to really understand the data, if we want the machine learning model to really understand the data, it must be able to know the boundary of the data distribution, the density of the data, what makes the data realistic. If you compare this simple example, if I show you this image and ask you if it's a real banknote, you can tell at a glance that it's not. But if I ask you to draw a very realistic banknote that can fool other people, it's much more difficult to do. So we believe the assumption here is that if we can train the model to mimic the data, the process of generating biological data or omics data, we hope that the model really understands the mechanisms and the biological knowledge that underlies the data itself. Okay, so a very simple approach to create a generative model, if you want to start somewhere, it's called an autoencoder. So an autoencoder is the idea that if you have a complex data, could be image, could be omics data. In prior class, we know that we can train a model to classify something about this data. Maybe we can want to classify that there is a bird in this image. The usual architecture is the image or the raw data is going to go into the machine learning model. It will get turned into some numbers, and then these numbers sometimes labeled as C for the latent factor. These are used to make decision, used to make classification. But in this case, because we want the model to be able to create the data, so we're going to use this representation, this embedding these latent factors, not to classify, but to create. So we have another machine learning model called a decoder. So this decoder basically decodes the embedding of this image and create another image that looks similar to the original one. So it's called autoencoder because you put in data and you ask the model to basically compress it, embed it, but then use that information to recreate the original data. You might think that this is trivial, but when we have the original data, let's say an image, it can be 100,000 pixels, or if we have an omics data, it could consist of 10,000 subgenes. But usually inside the model, this representation, this embedding vector, usually is less than 1,000 number. So basically the model has to find a way to turn tens of thousands of numbers, compress it into just a few hundred or a few thousand values that are good enough, sufficient for recreating the whole piece of information. So the model has to learn the ability to generate from some kind of a compression or representation. So the idea of generative model is we might not want to start with some data, we want to generate something new. Right? So we might want to give the model some numbers here, right? The vector c here. We can just sample it, right? Some numbers and ask the decoder to use our number and create something new. Right? It could be a new image of a different bird or a bird with different colors. Right? If it's a omics data, right, it might try to create the gene expression profile for a different type of cells, right, for data that we haven't measured. Right? It's not from the actual experiment. But the problem is, like, there is no guarantee that when you train this kind of model, like you put in thousands of omics data, maybe thousands of images. But when you sample a new numbers, like a random numbers, there's no guarantee that it's going to give you back an image of a bird, like, because not every number is going to map to the image of a bird, like, and not every number here is going to map to the realistic, okay, gene expression that you want. So in order to create a generative AI, like, we need to have some control over the way that the decoder learns the generation process. So there are several ways to do that, but let's start by discussing what would be a good properties, like, for a generative model. Right? The structure is simple. We want to sample some numbers, and we want the model that turns that numbers into realistic data. So we have basically two pieces that we want to optimize. First, like, we want to be able to sample this number, like, select the number that we want, and we have some idea that how these numbers will get turned into the image or the omics data of the cell that we want. Like, for example, you might want the data for a specific cell type. You want the data for specific disease. Like, we want to be able to control, like, the generation of the randomization of numbers here, so that we can produce the data from the class or from the condition that we want. Right? Otherwise, we have no control. If we have no control, then it's difficult for us to utilize the generative model. The second part is we want the quality of the data that the model generated. Like, when we tell the model to create a mutation profile of a cancer patient, maybe with a genetic background of Thai population, like, we want the model to have some quality that the data will follow our specification. Right? So if I were to summarize the property of good generative model, like, it should be realistic. Right? It should give us back a very realistic data. The way that we sample these random numbers to generate the data, it should be smooth, in a sense that if someone change your random number slightly, that person should get a very similar data. Right? We don't want the model to, when you adjust a certain number by 0.01, and then you get a completely different gene expression profile. Right? That's difficult to use, right? Because a small change can lead to a large difference in the output. Right? We want the model to be smooth. If we change the data slightly, if you change the input slightly, we should get a very small change or very similar data generated from the model. And the factor that we mentioned a few lectures ago, like, we want the process of the generation to be interpretable. We, as human, we want to be able to know when we select the value of C, right, what we are getting. Right? Do we get the cells from certain type? Do we get data from certain population? Right? We want to be able to interpret this process. So let's see how modern computer engineer overcome these design challenges to create the generative AI that we have nowadays. Right? So we're going to talk about some of these key techniques so that you get a sense when you read news article, like when you read papers, like you see these terms. These are the techniques that are being used nowadays. So after this lecture, hopefully you will now have a picture of what these techniques are doing to generate realistic data. So let's start with variational autoencoder. So we talked about autoencoder just a while back. We show that the autoencoder, the idea is you put in a certain type of data, and you ask the model to encode that data to compress it, and then use that information, the vector C here, to recreate back the original data. The problem here is if we do it in that design, we have no control over the parameter c. We don't know if c changed by a little bit, if you add noise to the vector c, how would that affect the output? Because the model can do anything. If we don't control that. So people who invent variational autoencoder try to add the smoothness property to the embedding space C here. So basically, when the model receives a data, when it encodes the data, instead of predicting just a single number, if you put in one data point, usually you get one number out or one vector out. But for variational autoencoder, because we want this embedding, this representation space, to be smooth, so before we generate the data, before we actually generate the data, we're going to add some noise. You see here, we're going to sample some noise and add to the vector, and then ask the model to generate. So basically, from the original data x, we ask the model to compress the data to get the representation mu. But instead of using mu directly, we're going to add some noise to mu. And we are still forcing the generator, the decoder, to be able to recreate the original data. So the generator here has to be very robust to this noise, because we're going to add different noise every time. But we're still asking the model to recreate the same data, because we use the same x. But we sample different noise, we get a slightly different C, but we ask the model to recreate the same thing. So the generator here is going to have some smoothness inside. When you change your C by a little, you're still going to get the same or similar data generated. So this is the first kind of very simple mechanisms that people add to the autoencoder design, so that you can generate data that have some kind of smoothness guarantee. And in fact, this design is powerful enough that it can help many problems in biology, as we will see later. So the idea of the variational encoder, just to recap, so we want the model to be robust to noises. So before we generate, we add some noises to the embedding, and we ask the generator to still reconstruct the original data, even though some noises have been added. But the problem is just a little technical problem. Who's to know, who's to guarantee that the model wouldn't set the sigma here, the standard deviation, to 0, and then the model can collapse back to the original autoencoder. Basically, one input gets one embedding and gets one output. So people have to do some engineering. Just if you're curious, when people train generative model, they are not just checking the quality of the final output, because that's depends on many processes inside the model. People are also going to check when the model generates the representation here, with the average representation and also the variance of the representation. They also want to force the model that this distribution has to look like some known statistical distribution. In some cases, we want the model to embed the data that resemble a Gaussian distribution. So instead of a single data point being mapped to a single vector, like a dot, in variational autoencoder, we are asking the model to map one data point, from our observation, to a Gaussian distribution, like this dash circular distribution surrounding that point. So instead of having for 3 data points, instead of mapping them to 3 representation vector, like a fixed representation vector, we ask the model to understand each data point as a probability distribution that centers around the embedding but has some radius, some variance. So this allows the model to be more smooth. Any point that you pick in here, the model is going to try to recreate the same data point. So this allows the model to sample kind of further away from the representation of each data point, but still give you back very similar result. So just to mention that it's not very trivial to build a generative model. Okay. But with this kind of variational approach, now we have a representation space that kind of smooth. Like we can guarantee certain properties of the output. But another limitation of autoencoder design is you need to have some data. You need to have some data. You have to have some omics measurement, and you're asking the model to recreate exactly those measurements. So the model is going to have some limited. So the technical term is expressibility. The model is not going to be able to express, like to create new things very well, because it's only asked to recreate the data that we have. Like we have a limited data, and we ask the model to be able to recreate just these limited data. So it has going to have some limitation in terms of what it can generate, what it cannot generate. So people try to overcome this problem. They want the model to be able to generate something totally new, right? Something that we haven't seen. Maybe it's a cell type that we haven't studied. Maybe it's an image of a virus that we haven't seen before. We want to be able to create something that's outside of our data. So people thought of this kind of adversarial design. So they call it adversarial because we're going to have two AI basically competing with each other. So we have the generator, which is the model that we want. Right? It's going to start from some random input that we picked, and it's going to recreate some realistic data that we want. But in order to train this generator, we're going to need one more AI, and that's called a discriminator. So basically the role of the discriminator is it's going to take the generated data or the synthetic data that we got from the generator. The discriminator is going to try to classify, trying to detect if these data are real or these are fake, like if these are synthetic. So the discriminator is going to take some output from our generator and also some actual experiment, right? Maybe from a different lab, right? And compare. It's going to try to identify these as real data and ours generated data as fake. So the idea is, like in the beginning, like we don't have a good generator, right? So the generator is going to produce something very bad, like something that's not going to look like a cell image. So the discriminator is going to easily say that, no, these are not real, like these are fake image. And that will be a feedback that goes to the generator to try something new. So the generator will try to update its coefficient to generate a different image, different kind of data. And as the generator learns to do better and better, the discriminator is going to have a harder time telling if it's real or fake. So the discriminator is also an AI. So it's going to learn as well that when the generator gets better and the data become difficult to distinguish, the discriminator is going to learn an even better way to distinguish the data. So it's now a competition. Right? The generator is trying to get better to create data that looks real, and the discriminator is going to get better and better at saying that these are fake. Right? So at the end, right, what people found that at the end of this kind of adversarial training, right, we can get a generator that's very good because it's going to produce images that are very difficult to distinguish between real data and their generated data. So this is a famous design because it doesn't need any new idea. Like you need some experimental data, but you don't use that to train the generator directly. Right? Because if you use these data to train the generator, you're basically limiting the ability of the generator to focus on this data. But instead, we are going to ask the generator to create anything that it thinks looks like the real image, and the one that decides whether the generated data looks real is the discriminator. So this allows the generator to create anything. It's not bound by our experimental data. It can create any type of realistic data that, as long as the discriminator cannot tell the difference, and the discriminator is well trained, right? These data are going to be really realistic. The data that we generated are going to be very realistic. So the idea is of adversarial training is a competitive learning process, because in order to get a good generator, right, we need to have a good discriminator that teach it, right, what's real and what's fake. Right? But the problem about adversarial training, right, is you can probably guess from the design, right, it's going to be very inefficient, because the generator doesn't really know anything about the biological data. It only learns through discriminator that what is generated is bad or good. Right? Knowing that the output is bad or good, like, doesn't really tell how to improve. Right? It's not similar to when it sees the actual data. It can tell how to optimize its generation to match the data. But in the adversarial design, it only knows that these are bad and these are good, but there is no details about how to make the bad data looks better. So these techniques, although the concept is very simple and it can be successful in certain situations, is very inefficient, right? And it's also unstable, because there's no real feedback, like, from the actual data into the model. So people try to come up with something more stable, something more deterministic. Right? And nowadays, I think you probably have heard of the term diffusion model before. Right? So it's a generative model that became very successful, I think maybe in the past 5 years, I think. So the idea is also very interesting. I want to introduce this. So if we want to generate, like, realistic data from noise, like, we want to sample some numbers and then we want to create the data from it. That's a very difficult process. But one thing that we can do, like, one thing that you can do very well, is if I give you a data, like if I give you a omics data, like gene expression profile, or if I give you an image, right, you can create noise from that image, from that gene expression profile easily. You can just add random values to your data. Right? If you add more and more and more random values, like, you are going to map your data gradually to a pure noise. Right? So this process is simple. Like, you add more noise, right? You let your data diffuse, like particle diffuse in the aqueous. Right? If you let all the particles move randomly, like, eventually it's going to be well mixed. Right? You're going to lose all the signal and you get pure noise. So people thought that, okay, if we can create, like, a process of adding noise, like, if you think of adding noise as a mathematical process, like, you add numbers to it. If you can turn from data to noise, like, with mathematical process, that means that we can train a deep learning model, an AI, that reverse that mathematical function. Right? So basically we have data, like, we add more noise, like maybe over 8 steps, like, until we get pure noise. If we can train the neural network, or if we can train a AI that learns, like, from through all of data, like, not just one image, but a lot of image or a lot of omics data. If the model can learn an inverse function that can map from pure noise to a less noise data, like, and then to a less and less noise data, we're going to have a model that can create a very realistic data from random numbers. So this is why this family of model is called diffusion model, because it's trained to reverse the diffusion process. Like, the diffusion process add noise to the data, but they're going to denoise, like, the data so that they get better and better quality output. So they call this a diffusion model. And just to show you some example, like, if you want to create, like, a realistic pictures of a dog, like maybe an Irish setter, a breed of a dog, the diffusion model, we try to start from maybe some random numbers, right, and then try to create. Like, first, it might get a blurry image of the dog, right, but still showing some realistic characteristic of an Irish setter dog. And then the model can try to reverse the process to create a better and better, more high resolution, even clearer image until at the end you get a very sharp, very clear image of a dog that looks like an Irish setter. And this is 5 years ago, so we have this capability. One thing that I want you to think about, if you want to imagine how this model works, we talk about the principle that from a single piece of data, one data point, the model is going to we're going to add noise to the data until we get very random, very pure noise, and then we're going to teach the model from different omics data, from different image and different noise that we arrive to at the end, how to reverse the process to get the high quality realistic data. You can think of this as a field of vector. If we sample some numbers in the diagonal, we can imagine that the model is going to take our randomly selected position and we move along the arrow until we arrive at some final destination. And at the final destination, there's going to be some data that got generated. So you can think of the process of this diffusion model as starting from a random position and then move along the vector field until it arrives at a certain point, and at that certain point it's going to correspond to some data that got generated. So this can be a mental image when you think about the generative process of a diffusion model. So diffusion model is quite good, because the process is very clearly determined. It's a noise addition process, and you train the model to reverse that. But it can be slow, because you have to add noise gradually at a time, small noise at a time. Because if you add a lot of noise, from this image, if you add a lot of noise directly to here, reversing it is going to be very difficult. So the key of diffusion process is you're going to add a little by little. You're going to add noise little by little, because a little addition of noise is easy to reverse. A large amount of noise is difficult to inverse. So one limitation of the original implementation of diffusion model is it's going to take a long time to run, because you have to gradually improve your data, your generated data, a little by little until you get to the end. So people are trying to think, this might be too technical, but it's a modern technique, so I want you to be aware of it just a little. So people want to make the diffusion process faster, instead of having to go through 8 or 10 steps, can we do something more easier? And when we map this kind of a vector field, from our sample the random numbers back to the data, to generate the data, can we make it easier to understand? If this vector field is easy to understand, you can just follow the arrow and then you get to that point. But some model might create a very loop, very big loops, very curved, vector field that can be hard to understand. So people want to try to also make the model easier for human to interpret. So nowadays, you might have seen some keyword about flow model or flow matching model. So the idea is with the stochastic, the noise, the diffusion, the addition of noise with the diffusion process, it can be very random. You can add noise in one direction and then change direction, because you add random noise. So the vector field that bring you from random numbers to the data can be uneven, can be in all over the place. But because the calculation that we need to clear the data, if we want the model to run very fast, if we can map the process as a linear function, if we know that from this noise it's going to walk in certain direction, for 5 steps, and then we get the data. From this random noise, we're going to walk in certain direction 5 steps, and we get the data. This is going to be very easy for the model to calculate. So people try to come up with so this flow-based technique is the idea that instead of using random noise addition, which can be as designed random, in random direction, can we make this noise addition, like a flow of water, like a flow of signal in a direct line, in a straight line, so that it's easy for the model to calculate. So this allows us to run generative model, like diffusion model, in higher throughput, because we can calculate the generation more easily. So before we go into the application in biology, I also want to show you some cool things that we can do, like to guide the learning of AI, and also including the generative AI. So you might wonder like how we can generate one type of data from a different type of input. Like when you type in, when you want to generate image, like you are starting from a description of an image. Like if we want a certain type of dogs in the image, maybe we want certain cell type in the image. You might wonder like how the models understand your language and associate it with a certain type of pictures. So there is a technique called contrastive learning. So contrast means you're going to compare something. So basically when we train the AI that does the generation, if you have a pair of image, like, and the caption, like, this image has the Aussie dog. Like, if you have this kind of pair data, you can train the model so that when the model try to create numbers, like to embed the caption, and when the model try to create the embedding, like, for the image, we want the model to map the corresponding, like, text and image to the same numbers. So basically contrastive learning, if we want to match, we want to pair data from different type together onto the representation. You can also think of this in terms of multi-omics. If you have gene expression data and you have mutation profile, like from the same patient, if you train the gene expression profile, like you get an embedding for the gene expression. If you train a model for mutation profile, like you get a embedding for the mutation. But if you want the model to understand multi-omics, the fact that this mutation profile gives rise to this gene expression profile, you want the model to embed like, gene expression and the mutation, like, from the same patient to the same number, so that when you sample that number, like the model knows this is a patient, like this is a person, and if you ask it to generate the mutation profile, you get that person's mutation profile. If you ask the model to generate gene expression, you get that person's gene expression. Like, so contrastive learning can help us map data from multiple modality, like multi-omics, to the same representation space. And these are the examples that we saw a few lectures ago. Like if we want to predict like, the properties of a protein, we have data as a sequence of amino acids. We have the domain annotation from interpro. Like we have protein-protein interaction from strings. But we can force the model to map these different modality of the data to the same number, like, using the same technique. Because for each protein, we know that this is a single protein, and this is the gene amino acid sequence for that protein. This is the domain annotation for that protein, and this is the protein integration partners for that protein. Right? So we can ask the model to learn these different concepts onto the same representation space. Right? So that's why we can use modern techniques to help us understand different type of data that we usually couldn't with bioinformatics before. Okay. And another example like that we saw earlier, like when we have multi-omics, like we can ask the model to map or embed like gene expression, DNA methylation, microRNA expression onto the same universe, like to the same space of representation, so that these can be used together easily. So this is also a magic of sort for modern machine learning techniques. We can start from different data type, but inside the model we're going to force the model that even if the model starts from different data type, we're going to force the embedding or the representation that we get to be compatible to be used to make prediction or to make generation downstream. This is something that we can force the model to do. In some cases, especially in physics, when we want to predict or when we want the model to generate certain type of data, it must conform to the law of nature. You shouldn't allow the model to predict something that violates the law of physics. So during the model training, when we put in the data, and we ask the model to generate something, could be prediction, could be generate synthetic data. Once we train the model, we can ask how does the output of the model depends on the input. We can calculate the slope, the gradient of the output related to the input. And if we know some some physical principles of the system, we have some constraint, something must be conserved. So we have some constraint that whatever the model learns, this output u, if we take the derivative with respect to time, with respect to x twice, double second derivative, these might have to sum to 0 because we know some property of the system. So people can use this as a constraint to the model that if the model is trained and this output u doesn't follow this law of physics, it's going to be some penalty. And if the penalty is high, the model is going to update itself to better match to this law of physics. So it's an encourage. We can encourage the model to follow some known mechanisms or known law. We can also guide the model indirectly. So this is guiding by the actual law that we know. But in biology, we usually don't have this kind of quantitative laws or mechanisms that we can follow. So instead, in this research, they want to embed single cell gene expression data to create a generative model. They want going to reconstruct using the variational autoencoder architecture. What they want to encourage the model here, on top of the usual variational autoencoder architecture, which try to make the representation space kind of smooth. In this case, they're going to try to match how similar cells, if you have cells with similar gene expression, you also want them to have a similar representation, because this representation should capture the original data. So in this case, they're going to create this network of cell-to-cell similarity. So these dots are individual cells, and we draw edges if the cell have similar gene expression profile. Once the model embed the gene expression for each cell into vectors, we can also calculate the similarity among cells. But instead of using the original gene expression, we're going to use the representation for those cells. So we're going to get two different networks. And the idea here is if you want to encourage the model to learn the representation that preserves the cell-to-cell similarity, we want these two networks, these two graphs, to be the same, or will be very similar. So in this case, we can also compare, as we train the model, we can compare these two graphs as the model change, as the encoder change, the graph for the representation will change. We can compare it to the original graph, the original networks between these cells. And this can be a constraint if the model create the representation that violate this network structure. It's going to be a feedback to the model that it has to follow this cell-to-cell similarity pattern. And in this case, it's going to give a better preservation of cell-to-cell similarity, which can be helpful for biological situation. Even if we don't know anything, we don't know any law about the system, but we know that if two cells have similar gene expression, we want them to also have similar representation, and we can guide them with by comparing the networks of similarity between cells. Another cool technique that we can do and it's helpful for biology, is called debiasing. The idea of debiasing is when we try to train a model, like maybe on certain gene expression data from certain cell type, like from certain experiment, there are always some bias or batch effect inside. So maybe you want to understand a certain disease, like a certain drug treatment, how it affects the gene expression inside the cells. But the different lab might use a different cell line from yours. So in their gene expression data, there's going to be an effect of the treatment, there's going to be the effect of the disease, but there's also going to be the effect of cell type that they use. And you want knowledge about the treatment, about the disease, but you don't want gene expression profile that occur because of the cell type, because you want to apply this to different cell type. So what you can do to debiasing techniques is, as we train the model to encode or compress your gene expression, the expression data, to the representation space, we, on one hand, we're going to use this representation to create, to generate, as usual, to generate recreate the gene expression data. But on the other branch, we're going to use this representation vector to try to predict, because when we take a gene expression, it comes from maybe a treated condition, untreated condition, or maybe from some cell type, maybe from some disease. When the model encode or create the representation for that cell or that sample, that representation can contain the information about the perturbation, about the treatment, about the disease. It can also contain confounding factor like cell type or cell lines as well. It all contained inside the gene expression information. What we want the model to do is we want the model to remember, to get the representation. That's good for the reconstruction, to create the gene expression back. But we don't want this representation to retain the data about confounding factor, cell line. So the idea is, when we train the model, we can ask the model to do multiple things at a time. We can ask this single model to try to recreate, reconstruct the original data. So that's the generative part. We can also ask the model to predict the confounding factor and also the treatment and the disease status. Usually, we're going to train the model to be able to do everything well. That's the usual target. You want the model to predict perturbation. You want the model to be able to predict covariance. You want the model to be able to generate. But in this case, if the model can predict covariance or confounding factor, that means information about the confounding factor is hidden inside the representation, because the model can predict. That means the information is there, inside the representation. So people thought of an idea that instead of training the model to be able to do this prediction of the confounding factor or covariance, how about we train the model to not be able to predict? And this is possible because we can when we update the model, instead of update in the good direction, we can reverse and update in the bad direction. So we can train the model so that it can predict perturbation very well, it understands the treatment, it understands the disease, it can generate very well, but it's no longer able to predict confounding factors or covariance. So this means that the representation space here, and this might take some imagination. So basically, if the model can predict perturbation very well, can generate very well, this means the representation here that we get from the data contain information about the perturbation and contain enough information to generate. But if we train the model to be poor at predicting confounding factor, that means that there is no useful information about confounding factor inside this part. Because if there are, the model would be able to predict. So we kind of purposefully train the model to be able to forget information about the covariance, but remember information about perturbation and remember just enough detail to recreate the gene expression. So this is a cool idea that is helpful for biology because we usually have many confounding factors that we cannot remove because the data is not designed to control every confounding factor. Like some confounding factor we have to kind of accept. So people try to come up with with techniques, with modern AI, like to make it forget about some of the confounding factor. But there's no guarantee. What I want you to take from this lecture is everything that we are doing here to train the model, like to train the modern AI, we encourage the model to do something and not do something. But we actually have no guarantee that the model actually remove some effects or if the model actually forget some effect. We only encourage it to do so. Okay. Another cool idea is when we want to generate, right? Sometimes you saw example like that we pick random numbers and we want to generate realistic data. We don't really care about what we want to generate. But in biology, sometimes you want to generate a very specific things. You might want to generate a microscopic image, but you have a very specific organization of the tissue that you want to mimic. You might have, for example, you might have a structure of cells, mitochondria, and background that you want this way. You want the data to look like this. So you can condition the generative process. Instead of starting from random numbers, you can tell the model that I'm going to generate, but I'm going to generate the data based off of this information. Please fill in the pixels for the mitochondria, like pixels for the membrane, pixels for the background, so that it fit onto this condition and it looks realistic. So the generative process can be conditioned. You can guide it toward a certain output. Okay. Another cool idea that we can do, similar to debiasing. In debiasing, we want to remember some information and forget some information. People try to expand that model further, and it's one of the examples that we're going to see in biology. Sometimes you have many confounding factors. Some are known. You know that the treatment dosage, the cell line, is going to be factors that contribute to the gene expression. But sometimes there are factors that you are not even aware of, maybe the protocol of the culture, maybe the batch of the reagent used, maybe those can affect the gene expression. Those are unknown. You cannot list out every factor that can affect your data. So people try to come up with this technique that they realize that in the data there are some attributes or some aspect of the data that we recognize, and there are also some unlabeled aspect. If you see the image of a person, you can tell the gender, you can tell the age, you can tell the skin color, you can tell the eye color. But there are other things that you cannot tell, but it affects the image. For example, the genetics of this person can affect his appearance, or the disease, if he's sick, it can affect his appearance. But these are not something you can tell from the image, and these are not even something that you can list from as factors. So people try to come up with this crazy idea that if you start from a data point, can be gene expression, can be image, some label that you understand, like cell type, treatment condition, we're going to try to have the model predict it from the data. So there's going to be some label, some attribute that we can ask the model to predict. Let's say in this example would be the gender, the age, maybe the race or ethnicity, we can predict from the image. But there are other crazy things that you might want to try. You might want to try to predict if this person is depressing, is having some kind of disease, or maybe mental condition. We want to predict, but we actually have no idea about the about the truth, because we don't have that information. The idea of this technique is we can ask the model to predict something that we know and also predict something that we don't know. But during the generative process, when we want to recreate this person's image, we're going to ask the model to trust both the attribute like gender and age that we know, and also these crazy prediction like disease or genetic factor that we don't know. But together, we're going to force the generative process to trust these two predictions. So the idea here is, if the model is able to create this person's, the people's image or cell or gene expression, by trusting both the known attribute and the unknown attribute, which can be erroneous, the model might predict something very incorrect here. But if these information are together, are good enough to recreate this information, the model must have gotten something right about these unknown factors. Because if the model got this unknown factor totally wrong, when it used this information to generate, there should be some conflicts in the output. So this is a very hand-wavy argument that maybe you can learn, maybe the model can learn about unknown factors that we human don't even realize, but they can try to infer it from the data. Okay, we're going to do some one example that we saw last time, and we're going to explain it again using this concept. Maybe it will help you understand. Okay, so let's see how these techniques, these concepts, help studies of biology. So just give you an overview. When we think about the roles of generative models in biology, we want the ability to generate. So for example, we can impute missing data, missing measurement, with synthetic data. So this allows us to explore conditions of measurement that we haven't seen before. We can create that condition, that measurement. We saw some techniques that might help us disentangle confounding factors. If we can remove it, then we can get to the knowledge in biology that can hold across datasets. We have seen the roles of these models to integrate multimodal data, multi-omics data, and obviously the ability to generate nowadays, we can use it to design molecules, predict 3D protein structure, predict chemical structure, and identify if certain drug and certain proteins we will bind together. Okay, so let's look at some example. This one I think we mentioned a little bit last time, but without the knowledge of this generative model design, it might be difficult for you to understand last time. But this time, now that we have seen some concept in generative AI, we can think about this problem more carefully. So in this case, we have single cell gene expression data, or any measurement from single cell. There are some aspects about the cell that we understand, what cell type it is, what tissue, what's the treatment condition, what's the time point. We understand this because we collect the data. But there's going to be some other hidden, these are unknown factors that can affect the expression level or the omics data that we are not aware of. It's not part of our experimental design. So people try to use the technique that we just saw. So the model, it's a generative model. It's going to take as input the single cell omics data. But at the same time, the model is going to try to predict the state of individual cells, which cell type, which treatment condition, which time point, and then use this prediction back into the model together with the data to determine both known attributes and unknown attributes. So these are some factors that we are not even aware of. But together, these are going to be used to condition the generative process. And we're going to train this whole system so that it can infer like something unknown about the cells. That can help in the generation of the realistic single cell data. And at the end, people that develop these techniques is going to analyze like for each cell what are the dimension of the unknown attribute that correspond to those cells, and try to make sense of the biological interpretation of these unknown factors. So this is one application where you can use generative AI to disentangle several factors from your omics data. And single cell data is a good match for generative AI because if you want to train model to generate, it has to understand the distribution of the data. And when you perform single cell experiment, you get data from thousands of cells. So the model has plenty of information to learn from. What are the possible distribution of gene expression inside each cell type? So it's a good, you will see a lot of application of generative AI specifically on single cell data. So in this case, they try to show that if you have maybe cells with known states, because you measure them, and you have unknown cells, you have cells that, but you don't know the status of the cell. You can use generative process in the previous slide to infer for these cells what would be their state, and impute the unknown state onto their data point. We can also try to create something that's not observed. In your experiment, let's say you might have a control condition, you don't treat it with anything, and you have a treatment condition where you try to infect the cells, maybe with the virus, or maybe you infect the cell with an empty vector. So usually you're going to compare control to empty vector to understand the effect of the experiment, and then to compare the empty vector to the virus and to understand the impact from the virus. So in this case, what we can do with generative model is we can take these control cells, which in real life we didn't treat them with anything, but with generative model we can try to treat them with either the virus or with the empty vector in silico, using the model to generate what would these cells look like if they were to be infected. So this is called counterfactual inference, because it's a what if analysis of something that didn't really happen, but we try to think what would the data look like if these cells are experimented upon. So in this case, we can see that from the original data, we can notice that maybe there are more than one cell type, or more than one status in the control. The data looks like it's not a single, might not be a single population. When they try to predict how these cells would look like when they are treated, they also found that there are indeed two populations. The cells that are infected respond in two ways, and the cells that are treated with empty vector also respond in two ways. So this might be one cell type, and this is the difference between no virus and with virus, and this is a different cell type inside the control. So using counterfactual inference, you can understand what will happen to the cells that were left untreated. You can also compare these predicted gene expression profile to the one that were actually treated with experiment, to see if there is any difference or discrepancy between them. So you can predict, you can create new data that wasn't there before. Another situation that's interesting, so when we analyze single cell data, we mentioned that we want to reconstruct the developmental stage, the developmental trajectory of the cell. We want to put arrows if these cells have developed into a different cell. But this is a difficult problem because we actually have no answer. We have to infer what are the age of the cells. So in this case, people use generative model that parameterize the time, like the age of the cells explicitly. So basically, this AI is going to take the gene expression data. It's going to try to predict the age or the time, like, of development for each cell. So they're going to predict this, even though we have no idea. Right? Together, they're going to also embed, like, the gene expression data, like, on using the variational autoencoder style. Right? And the pseudo time, like the age of the cells that are being predicted here, are going to be used inside the model, in the sense that if the model, like, infer the age of these cells, right, and we assume that the age or the time here are correct, right, if you sort the cells by age or by time, right, you should be able to model how the cell change over time. But if the model label the time incorrectly, right, and the model try to sort the cell based on this incorrect time, right, when it try to understand the progression of the cell development, like try to fit a function that mimics the progression of the cell development, there will be a lot of inconsistency because the times are incorrect. So in this case, kind of we assume that the model will eventually, at the end, kind of learn to create the time or the age correctly, and use that information as a self consistency check to make sure that if the times looks correct, it should be able to also reconstruct the development function of the cells correctly, and use those information to recreate gene expression data for every cell. Right, so this is a cool kind of generative technique that we want to infer something that we even ourselves have no idea about the actual correct kind of answer. But we can force the model to try to do this anyway. But once the model did the prediction, we ask the model to check the validity of the prediction in a certain manner. And if the prediction is correct, it should allow the model to generate gene expression data at the end that looks correct. But if the prediction of time here is wrong, the model should struggle to recreate the gene expression data at the end. So we use the generative property of the model to guide the prediction of factors that we don't know about. Right? A very cool application. People also try to apply this kind of a generative process to multimodal data. So let's say you have both gene expression and microRNA expression. Because we know that microRNA regulate gene by binding to the 3' UTR of certain transcript. Right? So people try to use this kind of cross-modality kind of interaction between microRNA and genes to train a generative process that goes back and forth between these two modes. So for example, right, if you start with messenger RNA data, because you observe the gene expression, you actually know something about the activity of microRNA. Right? Because if the gene is highly expressed, the microRNA is probably not highly expressed. Like, otherwise, it would replace the gene. Right? So you can train a generator that create. Let's start from the bottom here. So you train one generator that create microRNA expression, given that you know the gene expression. So you know the gene expression, if it's high or low, you infer the state of the microRNA. And once you infer the state for the microRNA, like, you can compare it to the actual observation, like to check if it's good or bad. So that's one feedback to the model. If it can understand the relationship between microRNA and gene expression, it should be able to use one mode to predict the other. So there are two generators here. One generator predicts gene expression from microRNA. The other generates microRNA from gene expression. And these two networks, these two generators, are going to work together, right, to help each other create a better and better generated omics data from the other modality. So at the end, we're going to have a model that understands very well how gene expression data tells us about microRNA and how microRNA tells us about gene expression. So essentially, we have a good integration between these two omics. And this entire model we can use to classify new sample. Maybe it's a disease that involves both gene expression change and also microRNA change. We want the model to understand the change in both modality together to make a single prediction. Okay. In last example, probably. So when we talk about protein expression, I'm not sure if I mentioned a little bit at the end in that lecture that when we move toward AlphaFold, from version 1 to 2, and then nowadays to 3, in AlphaFold version 3, one key feature is when AlphaFold 3 create the protein structure, it no longer use, so in AlphaFold version 2, it implements a lot of constraints from physics. Basically, it wants to guide the prediction of 3D structure that respect the law of physics. But that kind of design doesn't generalize very well to different molecules. Certain law of physics is good for controlling the structure of protein, but if you want to predict the structure of RNA, if you want to predict the structure of chemical compound, you need to come up with different rules that fit well with those molecules. So in AlphaFold version 3, they want to generalize their approach to be able to predict any type of compound, any type of molecules. So instead of using a specific handcraft rules, they turn to generative model. So in this case, they're going to start with random position of the atoms, and it's going to do something like a diffusion model to generate, to organize the position of the atoms together to create the 3D structure. Could be for protein, could be for RNA, could be for other type of compounds. So this is an application of generative model inside protein prediction. And if you go to that paper, you're going to see these diagram that tells you that the information about sequence alignment, information about distance constraint is going to point to this diffusion process to condition the generation. Like from random position, the diffusion process is going to move the position of the atoms, and the movement is going to be guided by the multiple sequence alignment information. It's going to be guided by the 3D distance information. So it makes sense that if you want to generate a protein starting from a random position, you want to condition, you want to guide the process using data from evolution and using data from constraint inside the generation. Okay. So those are example of generative approach and how they help the study of biology. So next week we'll talk about a different concept called foundational approach. And the reason why we need this is because of a simple reason. Like in biology, we have a lot of batch effect. We remember from single cell lecture that if you don't do anything to the data, you cannot combine, you cannot compare gene expression from different conditions or from different experiments. This is a big problem, because if one lab train their AI on their data, it's not going to be applicable to another lab because of this difference in batch effect. So the idea of foundational model is that we want the model that can be used across batches, across datasets. A simple approach is if you can combine all the dataset and you can train a single model. But the problem is one lab might study one disease, another lab might study a different disease. So you don't have a common target output to train the model. Different labs have different labels, different treatment, different disease, different population. So a foundational model, we want a model that can do this, the model that we can train on diverse dataset and is able to capture some generalized knowledge inside that can be used by anyone all over the world. you can imagine like you can have a model that you can use to predict maybe treatment or disease, like, and you don't have to do anything. You just copy the model and you can use it on your data. These are sometimes called like off-the-shelf model or zero-shot learning. You don't do anything. You just take the model from someone else and you use it on your data, and it works. If we can do this, then it will be very helpful for many biological labs. Or sometimes like we might have some data of our own, but it's not large enough to train an AI. If we take these foundational models that are built by large laboratory, we can fine-tune it, we can update it on our own dataset, which might be small, but it's good enough to tell the model that this is how our data is distributed. Sometimes this is called a few-shot learning. So we ask the model to look at our data for a little bit, and then we can use it for ourselves. Okay. So how do they do this? The big problem is different labs have different experiments, so they don't have the same label. You cannot train the model to predict cell type and expect it to work with every lab because the answers are different. One thing that you can do is you can ask the model to predict something about the raw data. So for example, if we want a model to understand a gene sequence that can work on every species, if we train a model on human, the model might not be able to interpret or predict the function of gene in yeast because it's different from human. But one thing that are common between human, yeast, mouse, and all species is we have the same DNA code. So there are two ideas that people can use to create this kind of foundational model. The first one is called mass training. So we intentionally remove some data, remove some piece of data, and we ask the model to predict what's missing. If it's a DNA sequence or amino acid sequence, you remove some of the amino acid or some of the nucleotide, and you ask the model to predict. If it's an image of a cell, you might remove some part of the image and ask the model to fill in what's missing. If the model can do this, that means the model understands how context or adjacent amino acid or adjacent nucleotide, how do these information determine the missing value, or in a cell image, if you remove the nucleus. If the model understands that this is a cell and it hasn't seen the nucleus yet, the nucleus should be filled in the part that were masked from the data. In a situation where the data is sequenced, gene and protein, sometimes people use what's called next token prediction. So it's similar to a language model. We ask the model to predict the next word. We start the first word and then we ask the model to fill in the next word, and the next word, and so on. In the case of gene and protein sequence, we can ask the model to predict the next nucleotide, the next amino acid. If the model can do this, that means it understands something fundamental about the raw data, which is true again, which is true for every lab. Anyone that analyzes gene sequence is going to underneath is all nucleotide sequence. So if the model understands these fundamental aspects inside the data, it should be able to adapt to any genes, to any species, to data from any lab. So this type of training is important because it doesn't depend much on the dataset, because we don't use any dataset specific answer. It's all about the fundamental distribution of the raw data. So for example, when we train a language model, instead of having to tell the model that this sentence, what does it mean, we can ask the model that, okay, I'm going to remove this word. You're going to look at the other words and try to predict what's here. If the model can do this correctly, that means it understands language. So, and this doesn't assume anything about the data. It just asks the model to predict what's missing. So these create models that you can adapt to different datasets, to different situations very easily. In biology, people have also tried this technique. Right? So in contrast to AlphaFold, to techniques that we mentioned in the protein structure lecture, where we want evolutionary information from multiple sequence alignment to guide the protein prediction. People found that if you train the model to understand amino acid sequence without multiple sequence alignment, so it just go through the list of protein, remove some amino acid, and train a model that can predict what are the missing amino acids. So the model don't really understand evolution. We don't feed it with multiple sequence alignment, but we ask the model to understand what are the natural proteins, because it can feel this alanine, this arginine. It understand that if it sees this kind of sequence, it must correspond to this protein. So the model can understand a little bit about what proteins are or what amino acid sequence are possible in nature. People found that this kind of training, even though it looks counterintuitive, it doesn't use the information that we as a human use, it still allows the model to predict 3D structure very well. It's not as good as AlphaFold because AlphaFold use multiple sequence alignment information, which is what should be used. But in this case, what they showcase is they can train this kind of model very quickly, because multiple sequence alignment, you need some computational power to process all billions of proteins to create the sequence alignment. But for this, you just take all the protein sequence, you just remove some amino acids, and you ask the model to predict. It's very simple to train, very fast. And what they found, which is very interesting, is this approach works better than AlphaFold for what people call an orphan protein. So orphan proteins are proteins that have no relatives. So these are proteins that even if you want to do multiple sequence alignment, it doesn't align to anything. So we see here that with the absence of evolutionary information, for orphan protein, we don't have evolutionary information. This kind of a very simple training design create a foundation model that can be adapted to predict the 3D structure for this kind of protein. In this example, people want to create a foundation model that can be adapted to any single cell transcriptomic data. So they try to train the data on millions of single cells from different tissues, from different parts of the body, from different diseases, to make sure that the model, if you remove the expression level of a gene, if the model can predict it, that means the model understands how gene expression from other genes can determine the expression level of the missing genes. If the model understands this, and this kind of gene-gene interaction, this is universal across every lab. If you study human, every lab is going to encounter the same gene-gene interaction. So if you train the model across all the tissue type or the cancer type, and it understand how to determine the missing gene expression based on the rest, the model can essentially understand the essence of biology of gene-gene co-expression. And you can adapt this model to any new single cell transcriptomic dataset. In this example, EVO2 is a foundation model for genomics, the first of its kind. It was trained on all reference genome, essentially all reference genome. And the training is you want to predict the next nucleotide sequence. So you give it the start of a gene, it will try to predict the rest of the gene until the stop codon, for example. The cool thing about this, which still requires some validation, is that even though the model only learned how to predict the next nucleotide, only that, nothing else is learned inside the model. When the researcher try to map, if the embedding, when you put in nucleotide sequence into the model, it will give you an embedding, how it turns the nucleotide into numbers. If you look at those numbers somehow tells you about the structure of the gene, even though in the training here, is only about the next nucleotide prediction. It doesn't even know what a gene is. But once it's able to predict the next nucleotide for every genome, somehow it learns some things about evolution, that this is a gene structure, and the intron and the exon are mapped differently. And here, in this bottom pictures, it tries to understand if the model can interpret the impact of a mutation. So in this case, it's taking a certain gene, right, and it applies mutation, so basically change the nucleotide. And it asks the model, does the model think the mutated and the original sequence, do these differ, or do they the same, or are they the same? Right? And in this graph, if the color is yellow, it means that the model thinks the mutation changed something about the nucleus, changed something about the sequence, like maybe some property or something. It's not trained in the model, but the model thinks something changed. But if the color is blue, that means that the model thinks that if you change the nucleotide, it doesn't matter. And again here, not only does the model understand the importance of the start codon, it knows that if you change the AUG in any way, it disrupts the entire gene, right? So it gives very high score for the start codon, right? And almost nothing in the UTR region. And inside the coding region, again, right, the model wasn't trained about the gene structure. It's only trained to predict the next nucleotide. But it turns out the model actually understands the period of 3 for the codons. The third position of the codon is the one where you usually doesn't change amino acid, so you can mutate and it doesn't create any effect. But the first 2 positions, if you mutate, usually you change the amino acid. So the model kind of is somehow able to see this difference in the importance of codon region by just looking at all the reference genome and learning to predict the next nucleotide. So this is a cool example of how when you train the model on raw data, somehow it understands the higher order structure of the data. It even understands some biology of the data, even though it's not trained to do so. Another example, for microscopy image. Nowadays we have a lot of digital image from many labs in the world. So if you pull this image together, you can train a model that understands every cell type, like every organelle, every kind of imaging device, like fluorescence, bright field, phase contrast. People train a foundation model for microscopy data to be able to segment many things, many objects inside the image. Okay, so to summarize, we nowadays we have 2 breakthroughs that help us understand complex data and to push science forward. The first one is generative approach. It makes the model understand the distribution of real-world data. It allows us to create kind of realistic synthetic data that we can use to impute, or we can use to analyze to make sense of the system. And the assumption here is that if you have enough data diversity, if the AI can learn to generate this data, which we probably agree by now is very difficult. If the model can do this, it must have learned to mimic some mechanisms that produce the data. So we can assume that even though there might be multiple unnatural ways to produce the same data, the way that the nature produces the data should be the simplest one. And if the model learns to do the same generation process, hopefully it should mimic this natural mechanisms. And we can use the AI now to understand that mechanism itself. And I want to contrast what we see in this lecture against usually supervised AI, the AI that make prediction that we see now, that we see more commonly. Because supervised AI only understand the data just enough to make that prediction, just to make classification, just to make prediction. It doesn't necessarily understand the whole distribution of the data. So there's some limited knowledge inside that it's not going to help you as much as generative model if you want to understand the mechanisms inside the data. We also look at the concept of foundation model, where you can train the model on diverse dataset, like to do very simple tasks, but somehow it has an emergent property that shows that it understands the higher order knowledge about the data, which is especially useful in biology. Okay, so that's kind of everything that I have for today.